Morning. Today we're going to be talking about stem cells in SCN2A related neurodevelopment disorders. And we're privileged to have Associate Professor Snezana Maljevic, who is a group lead at the Florey Institute in Melbourne in the area of Epilepsy Functional Genomics Group. Welcome, Snez. I hope it's okay I call you that because we have known each other for quite a while now and been working together in many different ways. If I can hand over to you to give us a little more background about the work that you do in general, but then obviously we're going to focus on SCN2A today.
Thanks, Chris. We've known each other for a long time. I'm happy to be here with you today and tell a bit more about stem cells and our work and SCN2A in general. So my lab is focusing on development of models that are based on patient cells, or so-called stem cell models. We are looking not only into SCN2A, but into different forms of DEE caused by different genes. My background is in ion channels and ion channel physiology. So majority of genes we study are actually either ion channels or channels that regulate how neurons talk to each other, so synaptic genes or similar. I've been a part of a larger team led by Professor Petru and have been working on development of some precision medicine treatments, including ASOs or antisense oligonucleotides. So that's basically a bit of a background. So SCN2A is one of the important genes we study. We have had several bigger projects that we're running in the lab with Steve and now separately in my own lab, where we're looking at the spectrum of diseases caused by SCN2A. And through previous projects and collaborations, we collected or were lucky to obtain a range of variants from patients from different diseases linked to SCN2A. So this is probably our largest pool of patients that we have available. Now, to study those, we use obviously very different models, and people have probably heard about organoids. So this is one of the major models we use. But we also have simpler models that enable us to look at initial development and how SCN2A is involved in creating how the brain will develop and what is linking the mutation that is found in patient to what happens in brain over time.
You've been doing that work across a number of years. You mentioned that you've developed these strong stem cell models for SCN2A. Could you walk us through what the key findings are so far from that piece of work?
Yes. I'd be happy to. We have started with a simple question. What is the difference between gain and loss of function or early onset and later onset DE? And can we see and map those differences to stem cell models? For those, we've looked into simpler neuronal stem cell cultures that can be developed within a month. Those are relatively easy to study. And we have looked at the electrical activity. So basically, do neurons fire more or less? Because more firing neurons can be linked to occurrence of seizures. When we checked the two mutations from each side of the spectrum, we could see that those cells modeled relatively nicely gain of function mutations. So we could see increase in firing rate. But for the loss of function, the findings are a bit less conclusive. So maybe the model is not completely optimal for that. There have been a variety of other mutations or patient lines we have tried. It is not easy, and I would just say that here, I know stem cells are really hyped and everybody loves talking about that, but the thing that we need to keep in mind is that we are studying cells that are developing, so the activity we see is much earlier than what happens in a child when they're born and get seizures. So it's like reproducing that aspect. The seizure aspect is still probably the most challenging part for stem cells, but what we did learn and what these models really did tell us and help us understand is how early the developmental changes are happening. Because if we use omics approaches in those patient lines, we can see that many genes change already really early in development, which means that this SCN2A, even though it has been treated as an ion channel that regulates how neurons talk to each other, fire, and how active they are, and how that can link seizures, And because we now know that it's not only seizures, there is also autism, there is intellectual disability, and all the other problems that affect patients, that actually could be linked to the activity or function that SCN2A plays already really at the very start of developing their development. And there is more and more evidence that these channels and these proteins we thought were only important later to regulate, how our neurons function, that they're actually also important in processes that determine how our brains develop. And that is why we are getting such severe phenotypes in patients. So I would say that we are, for SCN2A, understanding both, getting an understanding of which pathways are getting affected and understanding that the changes start really early in development. We have also recently started using a model that is actually showing us what happens within a first month of development in vitro. So it's still in a dish, but we can see that there are some morphological changes in our model that shows that initial form of brain development is already affected. And from what we have seen, the way that structure is affected at this earliest stage seems to be different between gain and loss of function and autism mutations in SCN2A, which means we could potentially use this model also to establish the genotype-phenotype correlation and have a stem cell model that can, on a very early stage, tell us, is it a gain of function or loss of function? And maybe if some of the gene therapies or other modulating therapies can be effective. I'd say probably the biggest takeaway is that SCN2A is a developmental gene. I know that we knew that because it causes developmental problems, but we now have evidence that it plays a role very early on.
Yeah, and just to ask a question, because loss of function is later onset, are you able to see that in that early stage?
Yes, we are seeing that loss of function, even though the seizures start later, the disease starts also early. So the changes start early in development, similarly to gain of function. They manifest through changes in different pathways. There is a bit of divergence in which genes and pathways are affected when we look at gain versus loss of function. Why the seizures come only later, we still don't really know. And we are now looking into maybe something that's not expected. Another unexpected role of SCN2A, or less expected, is that we usually talk SCN2A is, in brain we have a balance between excitatory, so the main neurons are the ones that are kind of guiding all the activity, and then we have inhibitory neurons or inhibitory pathway that sort of modulates how that activity, that it doesn't overdo. So like we know that the balance between excitation and inhibition in brain is actually how we all function, and that usually there is this disturbance of balance when we have seizures, but also other disorders like schizophrenia or other kind of neurological disorder can be linked to disturbance of inhibition, excitation. So it's when that balance is disturbed, it can happen in two ways. Either you increase the excitatory function or you decrease the inhibitory function. Both can lead to seizures, for instance. And we know that, for instance, SCN1A, so Dravet syndrome, is caused by reduced function of inhibitory neurons. And the SCN2A gain of function obviously increases the excitability, because SCN2A is the main sodium channel of excitatory neurons, and SCN1A is Dravet is main channel in inhibitory neurons. That's kind of the most simplistic version. But in recent years, we learned that there are also gain of function of SCN1A, which means inhibitory neurons should function more, and we still get seizures. And there are also loss of function of SCN2A, which means excitatory neurons should function less, but we still get seizures. So it's not so isolated and so simple. Both genes seem to affect both population of neurons. And we have just started a study studying the inhibitory pathway development, and we see some changes there due to SCN2A loss of function, which is quite interesting and something we're going to pursue.
Yeah, that's really interesting. For someone that has been in the healthcare sector, but not in the science world, I listen to yourself and others talk about this, the depth of research you need to find one piece and go deeper and deeper to really understand these complex conditions. It's never, one piece that you've done in terms of understanding potential treatments and cures, or, as you say, stopping it in that early developmental stage. So You're continuing to explore this. Are there any gaps in understanding that disruption in SCN2A, and how do you sort of see how we can explore that moving forward?
That inhibition pathway is one of those that has been explored, but only to a certain extent. And now there is more and more focus on that. And I think, as you said, it is a complex story. My work and the initial work in epilepsy research, when we had genetic mutations, was always in a very simple cellular model. Those studies did tell us something. You did learn that there is a gain of function, SCN2A, or loss of function, if you just use a simple cell line, put those genes in there and compare, and can say, oh yeah, this works more, this works less. We do have mouse models of different diseases, and we learn from mouse models, but it's mouse models. Developmental processes are difficult to assess. Over the years with new stem cell models, it's been also like a bit of a slower start, because everybody hoped they will resolve all of our issues. And, having a patient line and kind of human context is really good. But I think, the field is still struggling a bit with reproducibility, with, how do you translate what's happening in a dish to what is happening in the brain. But we are getting better there. And I think that what's going to probably happen is that we will learn more about the pathways that are affected. And I think that where we will probably head next is that there will be overlaps between all of these different DE genes, because we know that many DE genes, even though it's SCN2A or KCNT1 or KCNQ2, that there are some similarities in the presentation. When the seizures start, what is happening, and that there will be maybe pathways that we can modulate across different genes, not for each single gene separately. I would still say our biggest gaps are understanding what is actually happening during development, what is broken, and that difference between why is gain of function or why is loss of function linked to autism in some and late onset in others. These are big questions that we need to look into further. One other thing that is a big gap is that we still focus a lot on neurons. And we need to look at other cells within the brain, but also we need to look at other organs, because it's not the seizures, it's everything else. A lot of other things, from movement to gut issues to sleep. There's a need to kind of expand our focus, not from neurons and just like looking at what changes in activity, to what does neuroinflammation do? What do astrocytes do? What is happening in a gut? How is this affecting, I don't know, even heart? Like, what is the cycle, like daily cycle? Do we have changes in sleep, which we probably will find easy? So I think that should be our next focus. I think that's where the field should be moving. It's getting slowly there.
One of the things I really picked up from what you said then was really that sort of potential applicability across DEs, which I think is really exciting in the longer term. If there are ways in which some of the work that you and your team are doing is valuable across more genes than just the one. I mean, rare disease, when understanding can reach more of those rare conditions, then I think that is a real win for our community. I just want to turn our focus onto a piece of work that we as an organisation are supporting through a PhD that's going to start next year. Do you want to tell us a little bit about that work and how that will hopefully add to what we know?
Yeah. So first, thank you for that support. And we're really excited to start another project that will be a PhD project. And, the PhD students play a really important role in research. They come with new enthusiasm and energy, and they're ready to explore and learn. So the project specifically will actually look how those earliest changes that we see in that model that I said, the model of neural tube formation, the earliest changes in morphology, translate into an organoid development. So how does that morphological change that we believe is linked to the role of SCN2A really early in development actually affect formation of the cortex, formation of the inhibitory system? So can we actually, find a real proof what is happening there? And how are those pathways different between gain and loss of function variants? I guess with the idea to see, what would be the pathway. So, and when I say pathway, that means we always see there is one single mutation that the patient carries, right? But when we compare how many genes are changed in their expression in these models, we see that there can be hundreds of genes that are changed. So only one mutation and then hundreds of genes that are changed. That is probably partially due to changes in neuronal activity, which can cause the changes in gene expression on its own, because biology tries to compensate always. And if there are changes on one side, to balance that, there will be compensatory changes on the other. And also each of the genes is linked to some other genes and may play other roles. If something is missing here, it will impact what is happening in a completely different process. So what we are seeing in early epilepsy or in DEs is, for instance, that even though it's an ion channel and we are looking at the development of organoids, we will see that genes that are regulating how cells attach to each other are often changed. So cell adhesion genes, something that is kind of a different process than you expect, seems to be like commonly affected across different genes. We want to identify those pathways and those changes for SCN2A, DE, and that's going to be the main project. Obviously, the whole idea is that by identifying the pathways, we will have also the new drug targets. So we will know what should be modified. It is kind of a step beyond just fixing SCN2A, because obviously, yes, fixing overexpression. I mean, there are studies now and clinical trials that are going to modulate the expression of gain of function using antisense oligonucleotides. There are many small molecules that are kind of trying to modulate the activity exactly of SCN2A. But I think that what we want to explore is what is it that is changed when SCN2A is disrupted, that we can also maybe fix later in development or after birth.
That will be really fascinating to see that work within that study. Will you test anything? If it gets to the point, will you try and introduce things? Or I know that it's a PhD and there's certain time limits and capacity, but
we always try to have PhD students. I think that PhD students coming into our field are usually really motivated by the translational aspect and the fact that there is direct impact you can make by studying patient cell lines. If you find anything that helps in a cell line, there is potential to test it further. We definitely always try to have one aspect of the project that is translational. It's been very popular in mouse models to test drugs already available and see if some of them work. I think that would be the goal. In the first two aims, you're going to do the mechanisms and then you find your target, test some drugs. Because many of the pathways we see changes in, like I said, cell adhesion or how, cell attached to each other, for instance, or some of the developmental pathways. There are like many modulators that are already available, because these are pathways that are also like shared with cancer, and it's kind of like these are basic biology pathways, and there would be also available drugs from these studies. I'm not opposed to also testing some more targeted treatment like gene therapy or ASO treatment for other genes.
Yeah, no, that sounds great. Is there anything else you wanted to talk about in regards to your work? So I've asked my questions. So is there anything else that you want to share that you're working on?
Well, I don't know exactly about what I'm working on, but I kind of want to say it's been so important for us to have these interactions with you and with Geta and with parents. I just had a call yesterday with somebody from Germany, family from Germany. It was about KCNQ2 genes. So, we meet so many of you on your journeys. It has made such a change to the way we approach this work. I know the contributions we make are slow. Sometimes they look faster. Antisense oligonucleotides went relatively fast in how things are developing. We know that gathering all these little pieces of a puzzle does make a difference. And I'm pretty sure, that we have made such a big leap in DEs, like from understanding that there is a genetic basis to now understanding more and more about mechanisms, understanding that we need both mouse models and human models, from getting together, from natural history studies. So it's all coming into the big picture, and we need to know more about biology, and we need to be open-minded to test the things we didn't think of earlier. I think at least we'll have a chance to make some improvements in quality of life for you all.
Yeah, no, thank you. I have seen this real partnership between families or their representative organisations with scientists and researchers. That is an evolution because it really brings purpose and to that work.
Yeah, absolutely. I kind of wish that we were a bit better connected and that, government would see us more, or like that there would be a bit more funds available for these studies, because it's kind of, but it is as it is. We are all trying. And I think, it has real life impact. And I think that's really important and it's motivating us.
I just have one last question for you. You've started your own lab and you're building that. What are your hopes and visions for the work that you and your team?
Well, I do want to be a bit more brave and test some of the things that I spoke to you about. So move these things beyond just electrophysiology and testing the activity, and look a bit into maybe some other cellular types and some other organs, let's say, not only brain development. But I do see that vision of really understanding the changes in development that are linking these. So, what can we do that covers more than one gene at a time? I think doing and focusing on SCN2A and understanding, I think it's kind of all important. But when we did it across different genes and have those options of cross-linking and seeing, is there anything shared? I would like to go in that direction. So looking at shared mechanisms and early disruptions and what could be fixed later for more than one gene.
That sounds like a bigger grant proposal. We need to keep open for opportunities and support you in that. But I just want to thank you. Grateful for the work that you do, but it's always great to hear how it's progressing. And we look forward to working with you on that project next year and sharing that with our global community.
We're also excited about it. And thanks for the chat and for invitation, Chris.